Thank you all for being here. It's a pleasure to be here and have the opportunity to present Demeetra, our technology in gene editing. My name is Jack Crawford. I'm the CEO of Demeetra, and my talk today is called Undeniable CRISPR FTO, combining Cas-CLOVER IP with foundational CRISPR-Cas9 IP from the CVC. So Demeetra is a gene editing technologies company, and we provide targeted nucleases as well as transposases. And by far the biggest problem for all of our clients in adopting CRISPR-based gene editing for commercial use is the intellectual property issues associated with CRISPR-Cas9. So I'm going to speak about Cas9 as well as Cas-CLOVER intellectual property. I'm going to go over some validated commercial applications, as well as briefly touch on our licensing and access options as well. So first of all, what is Cas-CLOVER? Cas-CLOVER works in a similar way to CRISPR-Cas9 in that it uses a guide RNA that complexes with a Cas9, but the Cas9 is mutated to be inactivated for nuclease activity. So the guide RNA can complex with the Cas9, guide the Cas9 to the target site, bind the DNA, but it's fused to a dimeric nuclease called CLO51. The CLO51 being dimeric, you need both left and the right guide RNAs to come together at the target site, and the CLO51 is actually the protein that does the cutting of the genome to create your gene edits. So it has many of the benefits and then some additional benefits of CRISPR-Cas9 in that it has high efficiency, it's easy to use, you use the same guide RNAs as CRISPR-Cas9, but being a dimeric system, it lacks the off-targets that are associated with CRISPR-Cas9 and also lacks the translocations that occur. And it results in larger deletions with overhanging cuts, which results in higher knockout efficiency as well as higher knock-in efficiency. And I'll show some data on that later. But just a little bit about the intellectual property. When you look at Cas9, it's an extremely crowded intellectual property space. I use the patent database called lens.org. And if you search in title, abstract, and claims for Cas9, you get over 16,000 patents. So it's a very, very crowded space. It's one thing that I should note that also these foundational CRISPR-Cas9 patents, they don't expire until 2033. There's a similar patent scenario with other popular Cas enzymes. If you're looking to genetically engineer the genome with these Cas enzymes, Cas12a, you got 1,500 patents, small Cas9, a little bit less, and then another Cas is Cas-X. But in general, there's a very crowded intellectual property landscape. And so what does that mean for our clients? And any CDMO or pharmaceutical company doing cell line development is that the freedom to operate is uncertain, even if you have multiple licenses. Now, on the other hand, I just went over that Cas-CLOVER edits the genome with CLO51. That's our enzyme. It's a completely different situation. When you search for CLO51 in title, abstract, and claims, you get 30 patents. And they're from a small group of partners, including ourselves. So Demeetra has all of the exclusive licenses required for these patents to sub-license for commercial cell line development and bioprocessing. Our licensees also own all of their new discoveries and their new intellectual property that's developed. So if you make a cell line that has unique features and you think you want to patent that cell line, or you develop a new process using our gene editing technology, that's owned by you. And so you can see that CLO51 editing applications is a relatively untapped opportunity to develop new technology for your own business. So why did we choose to bundle our Cas-CLOVER intellectual property with the University of California, Vienna, and Charpentier, known as the CVC Foundational CRISPR-Cas9 IP? Well, first this is an overview example of how CRISPR-Cas9 works. You've got a guide RNA that complexes with the Cas9, and there are 2 active sites for editing the genome in 2 domains called the HNH domain and the RUV-C domain. And the CVC has a nice patent position on mutations in these domains, which cause the Cas9 to be ineffective in cutting the genome, but will bind and complex with the guide RNA at the target site, and then you can fuse to an effector domain. So that's an important piece of intellectual property for us. The CVC also has guide RNA IP. There are 2 types of guide RNA. A 2-part guide RNA system where you have basically the crimp RNA and the tracer RNA that anneal together, as shown on the left. And then you've got a single guide RNA, which is the 2 pieces that are covalently linked. And the CVC has IP covering both of those guide RNA setups. So why do we say that we believe we have a solution for this IP and FTO issue, and we call it undeniable FTO, is we have our whole system covered. With the guide RNA intellectual property, we have that from the CVC, California, and with the dCas9 IP, RUV-C and HNH mutations, that's also with California. The CLOVE51, we have the exclusive license from the inventor Helmholtz Munich, and this entire system is patented by our partners Poseida Therapeutics, and we have the exclusive license for cell bioprocessing. They use this technology in the clinic, so it's clinically validated for cell and gene therapy. And then Demeetra itself has also improved the CLOVE51 to make it more effective and have even more benefits. So the entire system, we can bundle this together in one license to our partners so that you can use this for cell line development with complete freedom to operate. So why does this even matter? The Cas-CLOVER also offers some technical benefits. It's validated in many different cell lines. What I'm showing on the left is targeted deletions at the glutamine synthetase, or the GS gene in CHO cells. We also validated it in plants, showing an albino knockout phenotype in tobacco shoots, as well as microbial cells, another red phenotype in this yeast that's been knocked out. So a little bit of data that we published recently with a group at Takeda is HEC293 editing for recombinant AAV biomanufacturing. And what you can see is we targeted the STAT1 gene, and in this figure, you see the deletion sizes are ranging from 10 to maybe 20, and some larger deletions in there as well. And the editing efficiency was over 92% with Cas-CLOVER, which is consistent with the highest values reported in the literature. We also confirmed that these larger deletions result in full knockout phenotypes. So we're showing the expression of the protein that was targeted, STAT1, and all of the clones that showed edits had no protein found in the cell. The one clone that was not edited as a negative control had similar phenotypes. So we achieve a disruptive knockout phenotype with our system. And then another example is what we call the clean-cut GS knockout CHO cells in combination with our harbor-in transposase. The Cas-CLOVER generated the GS knockouts in CHO-K1 suspension cells. We made stable cell pools with our transposase, which is completely license fee-free, unlike other transposases. And these data were presented to us by our partner at Hera Biolabs. And what we're showing on the left is the antibody titer for trastuzumab, or other known as Herceptin. And these are pools. and completely unoptimized in deep well plates. And you can see we got 4 grams per liter, up to 4 grams per liter in these pools. But what's even more striking is the cell-specific productivity, which is reaching 80 QP. So to finish it off, we also have innovative commercial access models. For Cas-CLOVER, it's our fields of use are cell line development, bioprocessing, and agriculture. We provide a technology license and a know-how license. And then we have an innovative single fee with no royalties license. And then we have special options for smaller CDMOs and biotech companies. And then for the Harbor Inn, it's a license fee-free system. And we'd be happy to talk to you about the different options. Thank you for your time.
Well, thanks, Jack. It's fascinating. You know, you've addressed most of all the criticisms I've heard, the limitations of the original system, so that's exciting. Now, do you have a booth here? We don't have time for questions.
We don't have a booth, but we do have 2 posters here. I think they're 60 and 63. Posters 60 and 63 if you want to come check it out. Thank you.